Very much for the invitation. I'm happy to be here talking to young scientists from Turkey and abroad interested in, computational biology, signaling, and systems biology. So today I will talk about how we can really understand and maybe even manipulate signaling networks, especially in the immune system, using computational modeling and single-cell proteomics. And before I maybe further continue, I'd like to mention that we are very interested here in the University of Chicago for applications both in graduate school PhD programs as well as for postdoctoral research from students and researchers like you guys. And please do contact me if you're interested in these positions. Okay, so starting from this cartoon of the famous NF-kappa B pathway. So what you see here on the screen is, on the left side, is the diagram, the network diagram, proteomic network diagram of the famous NF-kappa B pathway. This is, as you can see here, is the cell membrane. We have various receptors that detect molecules from the environment. These could be pathogenic molecules. These could be signaling molecules secreted by the immune cells, even physical stress. So these proteins are detected from the environment by the cell, and a big signaling cascade activates. And the information about the identity, dosage, timing, duration, and even spatial location of these signals are then conveyed to gene expression programs. So this happens by the physical translocation of transcription factors like P65, P50 complex, which is also called NF-kappa B, into the nucleus. And then once it goes there, it binds to the promoters of various genes and then starts synthesizing mRNA, which then lead to synthesis of proteins that will do various functions in the cell. So this is a very important pathway. It has been studied since past 50 years. There are 30,000 papers in PubMed on NF-kappa B, okay? And so it responds to environmental signals from pathogens, stress, own signaling molecules, and then regulates a bunch of different gene expression programs. And it is a very important pathway for understanding infection, autoimmunity, as well as cancer. It is involved in all these diseases. So NF-kappa B is an important signaling system. There are other famous examples like P53 involved in cancer, ERK, IRF3, TGF-beta, MSN2, and FAT4. These are all important signaling systems and transcription factors that essentially take information from the environment, convey this information into gene expression programs, okay? And in our lab, we have been interested in understanding how this information processing works from day one. This is a very interesting problem from an engineering perspective, also as a biologist's perspective. Now, we do worked on this problem for a long time. You can ask various questions such as, how do cells process increasing, decreasing, or periodic cytokine signals? Cytokines are these proteins that are secreted by immune cells. How do cells process pathogen inputs that are simultaneously present? What does the immune system do, immune signaling system do, when there is a bacteria in the tissues, there's viruses in the tissues and whatnot? How does NF-kappa B or other signaling pathways regulate gene expression in space and time in tissues? So these kind of questions are all very interesting and has been the topic of research both in my lab and as well as in other labs in the past. And oftentimes we use, as you can see here in the movie, live cell analysis. So these are individual cells expressing fusion proteins that target NF-kappa B pathway, and we can put these in different culture conditions, stimulate them with different ligands and signaling molecules, basically asking these questions in vitro, study them over time in live cell imaging experiments, and try to make sense of what might be happening, okay? So this has been a very rich topic of research, important both fundamentally as well as for applications. Now, there's an interesting point here, which is the pathway is complex. So you can see that there are a lot of proteins here, and this is already a reduced version of the pathway. So if one wants to understand how this whole thing works, for example, by overexpressing this particular protein, or maybe a mutation knocks out this protein, or maybe this protein, how does the whole system work in the different conditions like this with perturbations is a very difficult one, because the system is very complex. There is time-dependent phenomena, dynamics, there's cell-to-cell variability. So because of all these reasons, the pathway, NF-kappa B pathway, has been the topic of a lot of computational analysis. So from these diagrams, we can write down equations, starting from maybe differential equations describing chemical reactions here that are seen, and then solve these equations using a computer to predict what would happen with these perturbations in this entire system, right? So it's like a many-body problem in physics, and requires a lot of computational analysis because things are not very obvious how the pathway would change when you make different perturbations. So all these papers describe both experiments and computational analysis for regarding the NF-kappa B pathway. So this is the pipeline for both the experiments and computation. We, in our lab, not only do basic research in signaling, but also develop a lot of technologies, especially microfluidic technologies. As you can see, we have this device here that you can seed cells, individual immune cells, for example, stimulate them with different chemicals and ligands and signaling molecules, and monitor them over time with microscopy or other techniques, all in this single device in extremely high throughput. So you can see that the device is completely automated. This is like your well plate experiments. Imagine that these are well plate experiments where you're loading different fluids into the wells and then monitoring what's happening to the cells that are being cultured. But this is extremely high throughput. You have thousands of conditions generated in this device, all simultaneously and in a fully automated manner. So we make these kind of things. This is our research. It really enables also performing high throughput experiments with live cells in a fully automated manner. So this is what generates the data. And then we take data and then start creating models of the pathway, mathematical models, by really simulating what's happening in the cells. So here you have in the extracellular environment, you have signaling molecules like TNF, you have receptor signaling molecule binding, you have the activation of the kinase, transcription factor release and localization, and gene expression. All of these things can be combined or written down in equations and then simulated in a computer environment to predict what would happen. So there are a few things about modeling of signaling pathways or any kind of biological phenomena. We are talking here mathematical and computational modeling. Modeling oftentimes require a few things. One, you need to know the underlying structure. So that cartoon that I showed you, the diagram, is the underlying structure. Then you can write equations. But you also need the parameters to calibrate this model. For example, translation rates, transcription rates, translocation rates, binding constants. All of these numbers need to be experimentally obtained. And this is necessary so that the models are appropriately fit to the data and reproduce the observed things faithfully. If that can be done, properly calibrated models then can predict complex behaviors. So oftentimes we don't have either the resources or the capabilities to predict or do experiments that mimic what could be happening in vivo in the tissues, but we can run a computer and then make these predictions if we have appropriately calibrated models, okay? So models of NF-kappa B dynamics have been developed by others. I have been involved in this one since my postdoc times as well. So as you can see on this cartoon, starting from the TNF receptor binding to TNF molecule, this particular pathway here can be described with differential equations, basically describing all of these binding and activation and inactivation events. And ultimately, the equations are shown on the right side. Then you can, if they are properly calibrated, if you have all these numbers, then we can make simulations and then predict what live cells are doing. So on the left-hand side here, we have a very complex experiment. It's an actual microfluidic experiment where the cells have been put in culture. They have been stimulated with periodic pulses of TNF, this important cytokine, at different periods. As you can see, 30 minutes, 60 minutes, 75 minutes. These are repetitive pulses over time. It's like a pulse train. And then live cell microscopy have been performed. Every data set here contains a few 10,000 data points. So this whole experiment contains about 1 million measurements. And so we are measuring NF-kappa B localization dynamics. And this is a kind of a summary of the data. And this is our model. So as you can see, the model predictions reproduce experimental behaviors pretty accurately in every condition. So the dots are real experimental values collected from this big data set, and these are the predictions from the computation. So this shows that if things are properly built, you can actually simulate biological things, and you can get a lot of insight and ask questions. So the previous slide shows you only time-dependent events. So a cell is activated by a cytokine, transcription factors start localizing in the nucleus, gene expression starts, and this happens over time. Of course, in biology, things not only happen in time, but also happen in space. So, for example, when you have a small infection, and this actually is my own arm. This happened when we got the TB test, and people from Turkey, they all are vaccinated with TB. So when they give you this little prick here with semi-live TB bacteria, you create a reaction, right? So as you can see at the center, there's this tiny infection site. There is this bigger circle. So my arm is basically getting inflamed. And you get these concentric circles that are happening. So these are spatial phenomena that are about the spread of cytokine-driven signals in two-dimensional tissue, right? This is literally the experiment. Now, funny thing is, NF-kappa B is involved in this, this particular behavior. It is driving the cytokine signaling. And with appropriately calibrated spatial-temporal models, we can also predict these kind of behaviors in a computational model. So here's the simulation results. This is the infection site. We are predicting gene expression in space in this case. As you can see, signals are propagating for about 1 millimeter. And then they start expanding. And if you wait long enough, you are creating these gene expression zones, something in the center, a circle, and a bigger circle, and different genes are expressed in different locations here. This is the simulation result. And it looks very much like the actual experiment in a sense. So what I'm trying to say here is with good data and a good basic understanding of the underlying structure, you can extend computational models to space as well as time. Okay, so this is really pushing us towards better and better modeling and understanding of what's really happening when you are infected, when you're showing like autoimmune response, when you have vaccination. All of these can be simulated in a computer these days. Okay, so this brings me to data. Now, with modeling I mentioned that we are severely lacking data. Modeling approaches are fairly simple. You can use differential equations. You can maybe use deep learning methods these days. All of these are possible. What we are lacking is good quantitative data. So to address this problem, in the past, we have developed several technologies that generate the type of data that we need. An example here is this particular study back all the way back from our Switzerland times. So this technology is called digital proximity ligation assay, digital PLA for short. You take a cell, one cell, you lyse it in a PCR tube, and then you split the lysate into two parts. One part goes through the so-called digital PCR pipeline. In digital PCR, what you're doing is you first convert mRNA to cDNA, and then you take cDNA from the cell and then load it into small droplets. These are microfluidic droplets. There are 20,000 of them. And if you load them dilute enough, you will have a condition where most of the droplets are empty, but some droplets will contain the DNA from the cell coming from mRNA, okay? And when you do PCR amplification in these droplets, each positive droplet turns bright green, and the droplets that don't contain a template are dark, so you just count them, okay? And the machine can count it for you. This gives you absolute quantitation of mRNA counts in individual cells. Moreover, you can also take part of the lysate, combine it with these proximity ligation probes, and then run digital PCR on them to measure the proteins. So here's how it goes. You have individual proteins, two antibodies bind to them, and then each antibody have a piece of DNA that is specially designed so that when the antibodies are close enough in space, there's complementarity, and you're going to create double-strand DNA. And then you can take these products and amplify them with PCR and measure the protein abundance. So with this technique, very accurately, we can measure mRNA expression as well as protein expression levels with digital counting, all in the same cell. Now, what we did with this technique, we first showed that we have extreme sensitivity. We can measure very small amounts of mRNA and proteins, like all the way down to femtomolar. And then we look into the correlation between mRNA and proteins in the same cell. As you well know, mRNA is necessary to create a protein, but its existence does not necessarily mean that there's a protein in the cell, and vice versa. So these data points here are all coming from one cell. We are measuring CD147 mRNA copy, so on the x-axis, and we are measuring CD147 protein copy on the y-axis. This is 10 to the 5th. As you can see, if you look at the full data set, the correlation between the two molecules is actually fairly poor. So the lesson we learned from this is transcript counts, mRNA counts, is not a good measure for protein counts in the same cell, okay? A lot of experiments use transcripts, mRNA, as a proxy for protein, but this and many other studies in this area show that the existence of mRNA does not necessarily guarantee the existence of protein in these cells. If that was true, we would have expected all these dots to lie on this really nice diagonal, and the correlation coefficient should have been much larger than what we observe here. So using this, we can also develop a computational model of the central dogma in cells by using our data from the measurements and calibrating a model like this. So what we have in the model is gene can be turned on and off. These are the rate constants for on and off. Typically, it turns on by binding of a transcription factor. Then from the on state, we can create mRNA. mRNA can degrade. And then mRNA can lead to synthesis of protein, and protein can degrade. So when you use a model like this, you can write the equations and then use our existing data from these digital PCR measurements to calibrate the model. and then estimate the correlations of mRNA and protein. And as you can see, these are the estimates at different experiments. The circle is measured. This is the predicted. And we got pretty good agreements. So indicating that the correct understanding, again, the underlying structure and good data can lead to very accurate computational models. Okay, so this is an experiment and computational study of only one protein. And of course, in mammalian cells, like human cells, you have thousands of protein species that are expressed. And it will be wonderful to be able to measure as many proteins as possible to have a better understanding of the entire system. And perhaps we can even develop a fully calibrated computational model of these large-scale networks like NF-kappa B and many other signaling pathways, all in the single cells. Now, to do that, we developed a technology which we call proximity ligation, proximity sequencing. At this point, I'm going to talk about that. It's a proteomic technique. And the reason why we developed this technique is, again, we want to be able to do computation, understand how cells are processing information. They take inputs, create outputs, and there's something in the middle, right? The middle is the machinery of the cell, and we want to be able to model this. So to be able to do that, we have to have good data, and getting good data requires, in this case, a new technique, because mRNA measurements that could be produced by, for example, RNA sequencing will not solve this problem of having poor correlation between mRNA and protein. All right, so moving forward, if we have a model like that, especially if we can also measure protein complexes, then over time, with a technique like that, then we can model things and come up with accurate computational simulations. So to achieve that, we have developed this technique called proximity ligation, proximity sequencing. So the way it works is, imagine that you have a protein complex on the cell membrane. This is one protein, another one in purple. And we have two antibodies binding to each protein. And then we have these oligos specially designed. There's a region where with a connecting oligo, you can create double-strand DNA, and then synthesize this, amplify this, and then run this through next-generation sequencing, like Illumina sequencing, to get information about protein complex formation, okay? So this is all happening at the cell membrane at this point, because that's an easier target. So what we have in our assay is all these targets, probes. These are antibody pairs targeting different proteins. Each of them have a different barcode. This is the barcode design here, okay? So this gives us information that there was protein A here, there was protein B. There's a unique molecular identifier for digitization of the sequencing data, okay? And we develop all these probes, and then we put them on cells. And if there's a complex, we can get protein dimer complex information in the sequencing readout. Moreover, so everything is going into a single cell sequencing readout. From this, we can also estimate the number of proteins on each cell, as well as measuring the mRNA, up to 10,000 mRNA molecules, because everything is basically fed into a single cell sequencing pipeline like 10x genomics, okay? So the new thing here is the ability of measuring protein complexes, which did not exist before this technology happened. There's a second advantage, which is, imagine that I have n proteins that I'm targeting. N proteins can make n squared plus n over 2 protein dimers between each of them. So if you have 100 probes, you can make 5,500 dimer measurements, protein dimer measurements on each cell. So this is a massive improvement in terms of protein data collected from single cell measurements. And that's one of the advantages for maybe cell clustering and those kind of measurements. Okay, so this is our data. This is all coming from the genomic, or I would say sequencing experiment, but we are still able to predict protein expression. So this is an experiment where T cells and B cells are mixed. and clustered either using mRNA or protein signals. It works really beautifully. It's very accurate. We can also measure protein complexes. So, for example, in this cocktail, we have these protein targets, CD127, 28, so on and so forth. These are some important immune cell markers. And this diagram shows us the amount of dimer, protein dimer expressed or protein complex expressed between these proteins. So the highest is going to be CD9, CD9 homodimer. This is high, this is high, this is low. But basically, we get this two-dimensional data sets from each cell. And this is only like 8 protein targets, but nowadays we are performing experiments with 100 protein targets, leading to 5,000 dimer experiments, measurements. So these are single PBMC cells. This is a blood cell, like white blood cells. It's a cocktail of different cell types containing B cells, CD4 T cells, CD8 T cells, monocytes, and so on. And these, using TSNE or UMAP, can be fairly accurately clustered, both using mRNA labels and as well as protein labels. So in this data set, we have, in this particular published data set, we have 38 proteins, 741 protein complexes, and 10,000 mRNA measured from each cell. And about 9,000 cells have been measured. So this is a massive multidimensional multi-omic data set that enables us to dig deep into the functioning of these cells. Okay, so I'm not going to go too much into the details of the analysis, but what I will say here is we can measure macrophages, T cells, dendritic cells, and a bunch of other cell types with this. We can create machine learning models like logistic regression models and predict gene expression or protein complex formation from our data. And yeah, so we also develop like physical models of this, because there's actually non-trivial interactions between proteins on the cell surface. So you can have a protein complex that is true, like a stable complex, but also two proteins can come together transiently, and that can also lead to data. So that can lead to a signal. So we developed models and published them recently that simulate this physical scenario and estimate the backgrounds that we would get from this random ligation event. Okay, so this is all I will say about the published paper. Recently, we developed new methods. Hopefully, we will describe this in a paper soon, where we can also go into intracellular proteins and protein complexes, not only the ones on the cell surface, but also intracellularly, and measure various target proteins, which is very important. And moreover, we can also measure these things in a spatial context. So proximity sequencing can be performed using slides that have different capture regions on them. So this is an experiment where we use the commercial system from 10x Genomics, the Visium system, which has these spots. And each spot actually captures both protein through our proximity ligation readout and mRNA. And then each dot gives you a tremendous amount of information. And this is actual tonsil data. So this is a specimen from human tonsil. So this is a two-dimensional slice of tissue. And when we analyze it with our spatial proximity ligation, we can observe these different regions that show the expression of different markers from T cells, B cells, dendritic cells, epithelial and endothelial cells, all in a spatial context. So we can even observe the germinal centers in these tonsils. So this is a very important immune tissue, and we are now getting proteomic data both in terms of protein expression, protein complex expression, and mRNA expression from each spot in the tissue. Okay, so that's all I will say about this. I don't want to talk a lot longer. It's been about 40 minutes. And just summarize. So we are developing these new tools into a community source, increasing the multiplexing capability. Now we can measure about a few thousand protein complexes, moving into intracellular proteins, We can also measure post-translational modifications like glycosylation, going into spatially resolved manner measurements. And all of this is really to understand immune cell development in human immune tissues like the germinal center. Okay, so with that, I will stop. Thank my group who did all the work. This is an old picture from 2018. Pretty much everybody here is gone. We have new people, but nevertheless, it's a picture. We have these funding sources, which we are very thankful for. And then I'll stop here, mention that we are very interested in hearing from you guys in terms of PhD and postdoc applications. We have all these amazing projects. I'll stop here and take any questions.
Thank you, Professor Dr. Tai, for your precious and valuable talk. Now we can start our question and answer session if you want. Let's wait for those who want to ask their questions.
Sounds good. It was a bit fast. It was it talked about a bunch of different things. So I guess there might be questions, but there might not be a question too. Yeah, we'll see. Fatma Betul should be able to ask a question. Okay. Thank you, Nilay. I appreciate the comment.
Dear Professor, also we will share the presentation in RSG-Türkiye YouTube channel.
Okay.
Those who want to watch again, they can watch it in RSG-Türkiye YouTube channel.
Sounds good.
I guess it looks like there is no question left, or there is a question. It says, thank you so much, dear Professor and dear participant. Take care of and have a nice day.
Thank you very much. Have a good day.
Thank you.